Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The year 2000 witnessed the bestowal of the Nobel Prize in Physiology or Medicine upon Arvid Carlsson, Paul Greengard, and Eric Kandel, recognizing their seminal contributions to the understanding of signal transduction in the nervous system. Their work, spanning several decades and touching upon distinct yet interconnected aspects of neural communication, revolutionized our comprehension of how the brain functions at a fundamental level. This essay will delve into the genesis, evolution, and enduring legacy of their collective achievements, emphasizing the groundbreaking nature of their discoveries and their profound impact on the fields of neuroscience and pharmacology. The intellectual landscape preceding the work of Carlsson, Greengard, and Kandel was characterized by a nascent understanding of neurotransmission. Scientists knew that neurons communicated via chemical messengers, but the specific mechanisms underlying the synthesis, release, action, and inactivation of these neurotransmitters remained largely mysterious. The central dogma of biology, which outlined the flow of genetic information from deoxyribonucleic acid to ribonucleic acid to protein, provided a crucial framework. However, the intricacies of how a neuron's environment influenced its protein synthesis and, consequently, its function, were poorly understood. The concept of second messengers, small molecules that mediate intracellular signaling cascades, was beginning to emerge, hinting at the complex signaling pathways that lay hidden within cells. Arvid Carlsson's decisive contribution concerned the identification of dopamine as a neurotransmitter in the brain. Prior to his work, dopamine was primarily recognized for its role in peripheral physiological functions. Through meticulous experiments, Carlsson demonstrated that dopamine, specifically in the basal ganglia, played a critical role in the control of movement. He achieved this by demonstrating that the depletion of dopamine resulted in Parkinsonian symptoms in animals, mirroring the motor deficits observed in Parkinson's disease patients. Furthermore, Carlsson's research advanced in the development of L-dopa, or levodopa, to treat Parkinson's disease. This discovery had profound therapeutic implications, offering the first truly effective treatment for a debilitating neurological disorder. The development of L-dopa was a direct result of Carlsson's fundamental research. It highlights the rapid translation of his basic scientific findings into practical clinical applications. Paul Greengard's focus rested on the molecular mechanisms by which neurotransmitters exert their effects within neurons. He discovered that certain neurotransmitters, such as dopamine, act by triggering a cascade of intracellular events involving second messengers, specifically cyclic adenosine monophosphate, often referred to as cyclic AMP or CAMP. He identified the enzyme protein kinase A, or PKA, as a key target of the AMP. PKA, once activated, initiates a chain of phosphorylation reactions, adding phosphate groups to specific proteins, altering their activity and leading to changes in neuronal function. Greengard's work provided a detailed roadmap of the signaling pathways involved, revealing the intricate molecular choreography that underlies neural communication. His pioneering work showed that neurotransmitters induce changes by modifying the existing proteins within a neuron, often by changing the electrical properties of certain key proteins. Eric Kandel dedicated his career to unraveling the molecular mechanisms underlying learning and memory. He selected the sea slug, a Plisia, as a model organism due to its relatively simple nervous system and large, easily studied neurons. Using a Plisia, Kandel demonstrated that learning, specifically habituation and sensitization, involved changes in the strength of synaptic connections between neurons. He discovered that the neurotransmitter serotonin, when released onto sensory neurons, activated a signaling cascade that led to the phosphorylation of specific proteins, modulating the flow of potassium ions and thereby affecting the duration of the action potential. This, in turn, altered the amount of neurotransmitter released at the synapse, changing the strength of the synaptic connection. Kandel's work provided the first concrete evidence that learning and memory occur at the molecular level through alterations in synaptic strength. The direct applications of these combined findings were immediately felt across numerous fields. Carlsson's work on dopamine paved the way for the development of modern treatments for Parkinson's disease, substantially improving the lives of millions. Greengard's findings on signal transduction provided a fundamental understanding of how psychotropic drugs work, leading to the development of new and more targeted medications for psychiatric disorders. Kandel's research on learning and memory offered crucial insights to the development of novel treatments for cognitive impairments. The synergistic relationship between basic research and therapeutic advancement was highlighted in each case. Subsequent research built upon these foundational discoveries, the identification of dopamine receptors, and the detailed mapping of intracellular signal transduction pathways triggered by various neurotransmitters, became active areas of study. The role of protein phosphorylation in various neurological processes, including synaptic plasticity and neuronal development, gained widespread attention. Furthermore, the application of these findings extended beyond the realm of neuroscience. The signal transduction pathways identified by Greengard were found to be conserved across a wide range of cell types, opening up new avenues of research in areas such as cancer biology, immunology, and cardiovascular. The initial reception of these works was generally positive, particularly within the scientific community. However, the complexity of the molecular mechanisms described by Greengard's and Kandel's research, with its detailed biochemical pathways and the involvement of multiple protein kinases and phosphatases, presented a steep learning curve for many researchers. Some early critiques focused on the limitations of using a model organism such as a plisia to extrapolate to human learning and memory. However, these criticisms were largely addressed as researchers discovered that the fundamental molecular mechanisms underlying learning and memory were remarkably conserved across species. Initial concerns over the potential for the oversimplification of the complexity of brain function quickly faded as researchers made further discoveries. Several individuals and prior works exerted a significant influence on these groundbreaking contributions. Carlsson's identification of dopamine as a neurotransmitter built upon the earlier work of Julius Axelrod and others, who had elucidated the synthesis and metabolism of catecholamines. Greengard's work was directly inspired by the earlier studies on cyclic AMP by Earl Sutherland, who had demonstrated the importance of this molecule in regulating cellular function. Kandel's neurobiological approach to studying learning and memory owed much to the pioneering work of Santiago Ramon y Cajal, who had established the neuron doctrine, and to the behaviorist movement, which emphasized the importance of studying observable behaviors. The influence of these Nobel laureates' work extends profoundly throughout the scientific community. Carlsson's discovery of dopamine's role in movement disorders spurred research into the neurochemical basis of other neurological illnesses, such as schizophrenia and depression, and propelled efforts to develop more targeted drug therapies. Greengard's identification of protein phosphorylation cascades provided a fundamental framework for understanding a wide range of cellular processes and led to the development of an entire field dedicated to the study of protein kinases and phosphatases. Kandel's work on synaptic plasticity, as well as his use of advanced molecular techniques to study memory, has fundamentally shaped our understanding of how the brain stores and retrieves information. Their collective contributions provided a foundation for modern neuropharmacology and inspired generations of neuroscientists to pursue a deeper understanding of the brain. The impact on the field continues unabated.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.